Good morning, everyone. Thank you for joining the College of American Pathologists media briefing today on bird flu. My name's Robert Johnson, and I'm the Senior Vice President of Strategic Communications and Marketing here at the CAP. Today, I'm also going to be your moderator. Just a few things to cover before we begin. You've all heard this before, but we'll just go through it real quick. This meeting is scheduled to conclude at noon Eastern Time, so we have an hour set aside. It is on the record and it's being recorded, as you saw. We'll use that recording on our website later for anyone who missed the call, or if you want to download it for your own assistance in going back over comments and things. It'll be there shortly after we're done. We'll start with some brief opening comments from our panelists, and then our intent is to spend the bulk of our time fielding your questions. We want you to use the raise hand button, and that will help us determine the order of those questions. And if you don't know where that is under the react button, not the react button, but the react button in the meeting toolbar. We do ask that you please wait until I call on you before you ask your question. And finally, the staff will be putting the names of our experts along with their credentials in the chat for your reference. So to get our conversation underway now, I'm pleased to turn it over to Dr. Donald Karcher, who is the president of the College of American Pathologists.
Thanks, Robert. And I'd like to welcome you all to today's media briefing on the bird flu outbreak. I'm Dr. Donald Karcher, president of the College of American Pathologists. I'm also professor and immediate past chair of pathology at George Washington University in Washington, D.C. Pathologists are the physicians who diagnose disease and who direct clinical laboratories in the U.S. and around the world. In today's briefing, we're going to go over information about the bird flu and talk particularly about the important role laboratory testing for the virus plays in monitoring and hopefully controlling the outbreak and in diagnosing and treating patients who are infected with the virus. This week, the governor of California declared a state of emergency after the H5N1 bird flu virus was shown to have infected dairy herds in Southern California. California has also reported 34 confirmed human cases of the disease, mostly from contact with infected cows and mostly involving mild symptoms. Also this week, federal officials confirmed the first U.S. case of a patient with serious illness in need of hospitalization. This case was in Louisiana. The CDC and the entire public health community are closely monitoring the bird flu outbreak and taking necessary actions. The College of American Pathologists and the clinical laboratory community are also closely monitoring the situation should widespread laboratory testing for the virus become necessary at some point. I'll be joined today by two expert colleagues, Dr. Bobbi Pritt from the Mayo Clinic in Rochester, Minnesota, and Dr. Ben Bradley from ARUP Laboratories in Salt Lake City, Utah. Dr. Pritt will start by introducing herself and making an opening statement, and then Dr. Bradley will do the same. Dr. Pritt.
Thank you, Dr. Karcher. Good morning, everyone. I'm Dr. Bobbi Pritt, and I'm the chair on the CAP's Council on Scientific Affairs. I'm also a professor and chair of the Division of Clinical Microbiology at Mayo Clinic in Minnesota. I was the chair of the CAP's Microbiology Committee in 2020 during the COVID-19 pandemic, and I was also the director of the Virology Laboratory at Mayo Clinic during the 2009 H1N1 pandemic. So I've given a lot of thought to pandemics and how we manage them. Now, today's topic is H5N1 avian influenza, a form of influenza A. And this is the outbreak, as you all know, we're seeing among wild birds, poultry, and dairy cattle. I wanted to just quickly set the stage by giving some basic facts about influenza virus, because we're going to be talking about a number of terms, and you're going to see these terms in the news, and it can be a little confusing. So I'll start with the virus type. The two most common types of influenza virus are A and B. There's also influenza C and influenza D. A and B are the two most common to infect humans. Those are the ones we get vaccinated against every winter. H5N1 is a form of influenza A. Now, influenza A is further subdivided up into subtypes based on two proteins that we just call H and N. They stand for hemagglutinin and neuraminidase. They're proteins on the surface of the virus. And At this time in humans, the ones circulating for influenza A are H1N1 and H3N2. Those are the components that are in the vaccine that we all get for this year's influenza season. Now, H5N1, that's the subtype that we're talking about today, bird flu. Now, you could break it down even further. Viruses can be further categorized into clades and genotypes. That's because influenza viruses are RNA viruses. That means that they use RNA instead of DNA for their genetic information. This is inherently prone to errors during replication, so you get mutations which can lead to new variants, clades and genotypes. So I'll just finish by saying there are multiple clades. The current clade of H5 and 1 right now is 2344B that entered the country in 2021. It's been here ever since. And then of that current clade, the two genotypes that you'll hear us talking about today are the D1.1 genotype. That's the one that caused the severe disease in Louisiana. It was also responsible for the teenager severe case in Canada. So that's in North America. That's concerning. That's the one that's also seen in wild birds, but that's different than the genotype B3.13 that's in dairy cattle. So I'm going to turn it over to Dr. Bradley, who will talk a little bit about diagnostics. And some of the things he'll mention have to do with these types, subtypes, and the genotypes and clades. So Dr. Bradley.
Great. Thanks so much, Bobbi. And thanks everybody for joining this call today. My name's Ben Bradley. I'm an assistant professor at the University of Utah, as well as a medical director at ARUP Laboratories. We are a national reference laboratory. We provide testing across all 50 states. And so we've been very engaged with this H5 outbreak to monitor it closely. And I think the message that I kind of want to start off and share with everyone on the call here today is from the perspective of clinical laboratory testing. I think there's a lot of different groups that do testing, most notably public health labs, the CDC, but also our clinical labs. And what we're facing with H5 is a very different landscape than COVID. And I think that's kind of the first message I want to make clear to everyone on the call today. And that's for several different reasons. I think the most important reason to note here is that with this H5 outbreak, what we're seeing is that we actually have the capacity to detect H5 already in our laboratories for most labs. And the reason for that is the test that we use for seasonal influenza virus diagnosis, that flu A test, those flu A tests are able to detect this H5 subtype. What you're not going to see, though, is that it would give you a result of positive for flu A. It wouldn't tell you that subtype. So our assays can detect flu A, but not differentiate it as a H5 influenza virus. So that's the most important message here of how it's going to be a little bit different than COVID. The other difference, too, I think, is that we've been much more proactive in addressing this outbreak, in part because we've been able to monitor it more quickly. This isn't a novel virus like SARS-CoV-2 was when it emerged. And so the CDC has also been engaged with clinical and commercial laboratories to help prepare for H5. There were actually some awards that were issued in the fall for commercial labs as well as device manufacturers to begin building capacity for H5 tests. And then as well, some of kind of the regulatory issues that sometimes make it challenging to bring a test out have also been loosened by the FDA to help get us testing capacity out there earlier here. So those are just kind of my high-level messages here for our current testing capacity, but happy to talk about any of that in more detail as well.
Okay. Thank you, Doctors Karcher, Pritt, and Bradley for those opening comments. We are now available to take questions, and you can again raise your hand by clicking into the React button at the toolbar and raising your hand there. We also have a question that was posed in the chat. So why don't we take the chat question first? I would like you to, unless you're just incredibly shy and don't want to say it verbally, we would, in order to get everybody in the order that they raise their hand, if you could use your hand button, that'd be great. So just the question in the chat for the group: might the different subtypes explain different clinical symptoms? And in parentheses, conjunctivitis versus respiratory distress, for example.
I could start with that and then I'll turn it over to Dr. Bradley. But the subtypes would be H5N1, H1N1, H3N2, and they do have different clinical associations depending on the strain that's circulating. But I think the question you're really asking is it the genotype? That's where we're starting to see different clinical appearances, perhaps. We haven't had a lot of human cases. But yes, the genotypes, based on those mutations that occur in the virus in H5N1, from the small amount of data we have, do seem to be associated with different clinical appearances. So the D1.1 genotype is the one we've seen in the 2 human cases that have been severe, and that's also the genotype we see in wild birds. Now, again, we only have 2 human cases, so it's yet to be seen if that genotype is going to be more associated with severe disease. And then the B3.13 strain is the one we're seeing in dairy herds, and the humans that have been infected with that have mostly had the milder symptoms like conjunctivitis. But it's interesting because we have had a viral isolate. They grew up the virus from one of those B3.13 genotype strains, and then they analyzed it in the laboratory and then tried to infect laboratory ferrets with it, and it was 100% lethal. And that particular strain looked like it had acquired mutational changes that might have allowed increased airborne transmission. But the thought is that was probably limited to that one person. But that just goes back to why it's so important to monitor, because these are RNA viruses. And when a virus is replicating within a person or another animal, it can have ongoing mutations that could make it more easily to pass from one person to another. So at this point, the B3.13 strain doesn't seem to be associated with severe disease, but we'll have to keep an eye on that. And Dr. Bradley, anything you want to add to that?
No, I think you covered that great, Bobbi. I mean, one of the good outcomes from COVID has been that we've really seen an expansion of the availability of next generation sequencing platforms available to do rapid genotypes of these viruses. And so we've been able to monitor very closely certain mutations that are present in the virus to look for patterns that show that it's beginning to adapt to humans or mammals from birds. And right now, for the majority of what we've seen there, we haven't really been seeing those adaptations that make us worry that it's getting more cozy with humans versus, say, wild birds in those situations. But certainly mutations in any of the proteins for the virus can change its clinical presentation. At this time, I don't think there's been any clear data showing why some of these initial cases were found with conjunctivitis versus respiratory symptoms. It could just be by virtue of kind of the path of infection in these farm workers, in these poultry farm workers and dairy workers, where either there's splashes or kind of material that gets in the eyes in those situations.
Very good. Thank you for that question. Our first hand is Adriana Rodriguez. And can you just, before your question, tell us who you're with so that our panelists know that?
Absolutely. Sorry, I should have done that in the little Zoom thingy. I'm with USA Today. First and foremost, thank you so much for taking the time to do this call. I'm sure a lot of people have so many questions about this. Dr. Bradley, I appreciate you addressing why maybe we shouldn't be so concerned in terms of like the parallels of like the COVID pandemic and whatnot, how we weren't ready before, how we're more prepared now. But I was wondering if maybe you should, or if you could address maybe, what are signs in which maybe we should start getting worried that this could be sort of an outbreak among humans? I know you
Yeah. No, that's a great question.
But,
So I think there's a few things that do concern me, and it's been, I think at this point now, 2 cases in the US we've seen where there's not a clear link to dairy cattle exposure, poultry exposure in these infections. I think it was a Michigan case that was first, and then there was a case that was found in California as well of a child without known exposure. So those things are certainly, concerning for me, because we would like to see that there's a clear epidemiological link to a bird or poultry or something like that. So if we would be seeing more of those cases, that's something that would lead me to be concerned. Another concerning feature to me as well is some data that recently came out from a serology survey. So serology surveys, we kind of look for antibodies against H5 virus to say, okay, this person isn't infected right now, but sometime in their past got infected with this virus. And what the results of that study showed was that there probably are more people getting infected in farms, dairy cattle workers is I think who this was done in, more folks getting infected than we're detecting right now. So even though we say we're at 60 some odd cases, that real number is higher than that. And so not quite knowing the full picture of things right now is always concerning too. But we do have some tools that we can use to kind of monitor for this. I mentioned the sequencing. That's one of our really big tools. So if we're seeing more human adaptation, that's going to raise red flags to say, all right, this is something we need to be more aggressive in testing for. And then something else we can look for too is just kind of basic trends of flu levels. So, over the summer, people don't get flu in the summer. So if we saw a big uptick in the number of folks getting diagnosed with flu, that could be kind of a red flag for us that something's out of sync here. We need to start investigating this more. Now, it's more challenging if you have these kind of peaks overlapping in winter to separate that out. And that's kind of what some of our surveillance testing is done for. The last comment I'll say here too, and kind of challenges for surveillance, which I realize maybe wasn't your original question, but we are dealing with kind of marginalized populations here, folks whose livelihood depends on them going to work and not being sick. So that also raises challenges for testing this kind of vulnerable population. And that's something else that we've really needed to, from a public health standpoint, kind of make efforts towards testing.
Thank you. I'm sorry, just a quick follow-up for clarification. That second thing that you mentioned about possibly more transmission that we're not catching, is that sort of leaning into, is that a nod towards asymptomatic transmission possibly that we're seeing?
Yeah, I think it's probably a combination of a few things. It may not be asymptomatic. It may be kind of subclinical. So not enough for somebody's like, I had a cough for a few days or my eye kind of felt bad, but that wasn't enough to get me to go to a doctor. I just kind of thought it was, whatever, a bad day. Or again, folks who just haven't wanted to go to the doctor for reasons of not wanting to miss out on work or something like that. But yes, these individuals probably had either asymptomatic or subclinical presentations.
Thank you.
That's a good point, Dr. Bradley, too. I'll just add, we talked about the 61 confirmed cases as of just a couple days ago on the CDC's website. That means confirmed by the CDC or public health labs. But there were some people that they think probably definitely had infection. It just never was confirmed. They weren't able to catch it at the time of infection when a PCR test would be positive. And then you have all those patients Dr. Bradley talked about that had that antibody serologic evidence of past infection, but were never tested. So I like to say that the 61 cases is probably the tip. I don't know if it's of a big iceberg or a little iceberg, but I think what we can say is that the 61 is going to underestimate the true number of people getting infected. We just don't know by how much.
Let me also add that California has 34 confirmed cases. California has a very robust public health testing program. Not all states have an equally robust program. So it's very likely we're missing discovering cases in other parts of the country because of that.
Excellent. Let's take our next question. Carter Williams, you were up next.
Hi, yes, Carter Williams with KSL.com in Salt Lake City. This one, I guess, is for whoever can answer it, but obviously during the COVID-19 pandemic, we saw a lot of mutations with time and everything like that. How has this flu mutated since the outbreak began? I believe it was in 2022. And then how many strains are out there right now?
Dr. Bradley, do you want to tackle that?
Yeah, so I mean, I think this is it, maybe a challenging question without getting too far into the weeds there. But you know, in every person that gets infected, there are small mutations that happen. It's just a question of, is this, and it's all by random chance. So this thing is just kind of throwing out random mutations to see maybe this works better, maybe this doesn't. So you know, the virus is constantly mutating, but there are occasionally mutations that come out that give it a little bit of an advantage in either replicating or spread between people or evasion of the immune system, evasion of antibodies. And as far as kind of what we've seen from 2022, 2021, when this clade 2344B kind of came on our radar, there have been some mutations that have occurred just to kind of show its evolution, but those mutations that it's acquired so far in terms of what it would mean to human pathogenicity is really unclear. We sort of historically and from in vitro data, we know kind of some high risk things that we worry about. So there are some mutations that can happen in the hemagglutinin gene, the HA gene, where we get that H5 from. That HA gene is what's responsible for it essentially binding to our cells and getting in. So we know that there are some mutations that can happen there that make the virus better able to infect our airway as opposed to the gut of birds, which is where this virus is typically found. And so that's one that we monitor really closely. There's a few other changes we can see, but like I said earlier, so far we haven't really seen those high risk mutations that we know are kind of concerning for increased human spread or for increased transmissibility pathogenicity.
So just to
Really?
follow up to that, the case in Louisiana, is there a specific thing about that strain that led to that being a little bit more serious than other cases?
Yeah, I think we don't know the answer to that right now. The case from Canada, which was the same strain as the Louisiana case, was sequenced and that's been analyzed. But to my knowledge, there hasn't been sort of some kind of linchpin found here that would say, like, this is exactly why this happened. Typically, you can see sometimes more severe infections with these spillover events from animals into humans, just because the virus hasn't necessarily adapted to humans yet. And in general, as a virus adapts, it kind of becomes less pathogenic in hopes of being able to spread better. We've seen that to some degree with SARS-CoV-2. So this is kind of I'll say maybe a prototypical picture of like a zoonotic event where a virus from an animal gets into a human and someone can get pretty sick from that. But there isn't a known mutation that's explaining this right now.
Yeah, not just Dr. Bradley said that so well. I'll just add in that there's a couple things that are worrisome about this, but it is true that at this point, there are no mutations that are really linking this to, that are making us worry that this is going to widely spread between humans. But because there are so many animals infected in every host, within that host, the virus replicates. And as it replicates, it has mutations that just increases the likelihood. The more virus you have out there, the more that's replicating, the more mutations could occur that would then make it more transmissible among humans. And then the other thing that worries me a little bit is that it's also infecting such a wide range of mammals, including domestic cats, cats as pets, pigs. Pigs are an important one because pigs can get infected with bird influenza viruses and human influenza viruses. And then you kind of have like this mixing pot where the two viruses can share genetic material and create new viruses. So that's always a concern as well. Hence the need for continued monitoring, even though at this point we're saying no need to panic, and we have tests that can detect influenza A, but let's keep a close eye on this.
me just add to really emphasize that right now there is no evidence of widespread human-to-human spread of this virus. There are a couple of cases where there's some question, but nothing definitive. And the severe illness that was in the case in Canada and recently in Louisiana is a genotype that's common in wild birds, which means it's not isolated to dairy herds, for instance. So that does offer the potential for this to spread if people are in contact with wild birds.
Thank you. Next question comes from Bob Riddell, I believe it is. Bob?
You got it. Thank you. My name is Bob Riddell. I'm a reporter at NBC Bay Area here in the San Francisco area. And Dr. Karcher and Dr. Pritt, I think you pretty much answered my question about whether there is documented human-to-human transmission. So my next question would be, based on what you're seeing and based on what you know about how these viruses mutate, how likely is it that we would eventually or could see human-to-human?
Pritt, I'll let you start with that.
I wish I could look into the crystal ball and predict that. I can't say that I can predict how likely this is, but as I had said just before this, it makes me nervous seeing so many animals and so many different types of animals being infected. That, in my mind, increases the likelihood that we could get a mutation that would result in more human-to-human spread. But I can't put a number on it. Maybe Dr. Bradley wants to predict.
No way. See, you guys are not going to get me on recording saying this. But here's what I'll tell you how I think about it. I originally grew up in South Louisiana, where we have hurricane season every year. And every year, you don't know if it's going to be a bad year or a good year. And even once a storm forms, you see all these spaghetti models of maybe where it can go or where it can't go. And so I'll say, this pandemic influenza threat is something that will always be with us so long as there are waterfowl on this earth and so long as there are mammals. This is something that's always going to happen. And these events are not going to become less over time as we get, more interface between humans and animals, more international travel. These events become more likely. So is it possible that a year from now, we'll still just see maybe minor circulation in herds and that this never became something that required a high degree of testing in humans? Yes. But at the same time, as Dr. Pritt mentioned, really it's just kind of one pig away from becoming maybe a big threat, where you get an infection of a human virus and this H5 virus in a pig. The viruses reassort. Their genomes are segmented, so you can think of it as like a deck of cards. And so whenever two different decks of cards infect the same animal, you shuffle them together and you pull out something new. And so we're now in a situation where those events could be happening. So putting a timeline on it, I think, is challenging to say, but I think we can say we're in a situation where we need to be alert to the possible threat of this.
I will add that they are working on an H5 vaccine for dairy cattle, and that would be helpful in decreasing the total number of animals infected. It won't help the wild avian fowl, the birds that are circulating. They're going to be migrating around the world, and they're always going to have their own influenza A viruses. So that will remain a threat, but at least we would get it perhaps out of the dairy cattle that may be leading to infections of some humans and maybe some other animals.
Yeah, let me also remind us that the previous coronaviruses outbreaks didn't always lead to widespread disease in the past, until the virus that led to the COVID-19 pandemic developed very high infectivity capabilities for humans. So but as was noted, this is a random process that viruses undergo, and it's really hard to predict when that event's going to happen.
Quick follow-up, would we be talking about this specific bird flu virus if we weren't just coming out of the COVID-19 pandemic? I guess because I know we've had these before, but I don't remember there being this much attention. So is the attention being driven by us in the media because we just came out of a pandemic, or is there something unusual about this bird flu virus compared to other ones that's driving the coverage?
So I'll start out by saying yes and no. Yes, we probably are much more sensitive to viral outbreaks because of the COVID-19 pandemic. But no, this is not new, and we have paid very close attention to previous outbreaks of influenza viruses prior to the COVID pandemic. So this is not new for us.
I think what makes this a little, yeah, I think
Yeah,
I'll add, I agree completely what Dr. Karcher said, and I'll add a little bit that what seems to be a bit new with this is just the large number of animals that are infected, particularly in North America, where there's concern that we might become the epicenter of a mutation that leads to an outbreak. And so it really falls upon us and our public health labs to do this close monitoring, the USDA to test milk and test animals. That's what makes this different in my mind from the previous H5 outbreaks that we've seen that have been a little bit more confined.
Thank you.
I think that is a good follow-up or lead in to the next question, which is on the chat. I would just remind everyone we've gone through every hand that's been raised. So if you do have a question, just go to the React button in your toolbar and raise your hand, and we'll get your question in. So from the chat, the question is, do you expect the test to work even if the virus were to mutate and adapt to human transmission? How much mutation would you need to see for the test to fail?
So I'll kind of jump in on this one first, and I'll say, so for our influenza A tests that are able to detect H5 and H1 and H3, H1 and H3 are our seasonal strains. The majority of these assays target a very conserved region in the virus, something that's essentially we have found to be essential for its function. And if mutations occur there, the virus really doesn't work as it should. And so that is what the majority of these flu A virus assays have targeted. So we feel like it's pretty strong, it's pretty robust, it works across many different subtypes of influenza A. So for that kind of base influenza A assay to fail, I think it would take a very significant mutation that we haven't seen before, that hasn't really been supported in the literature as being something that happens when it adapts to humans or even in wild birds. So that's kind of part of it. The other part is that H5 specific target for a lab that may be developing an assay that tells you, is this H5, yes or no? So with that one, we certainly need to be more careful, because that H5 specific assay is going to use targets of that HA gene, and that HA gene tends to have higher mutational levels. So one way we can address that is, again, by this kind of continued surveillance and looking for new mutations to emerge. Another way we do it when laboratorians build these assays is we typically take a belt and suspenders approach to designing an assay. And what I mean by that is we'll often use 2 or 3 targets that are specific to the analyte we're looking for, to make sure that if we do have a mutation that happens in one of those targets, we've got another 1 or 2 targets as backup to ensure that we're still able to get a positive result. And this was something that we saw with COVID as well, where we had N gene dropouts, we had S gene dropouts that happened over the course of its evolution.
Yeah, so I think the good news is we would be able to continue to detect the virus. Let's say it mutates H5 and starts circulating through humans. We could probably say with confidence we could still detect it as influenza A. For some of those tests out there that actually tell you if it's H1 or H2 or H5, or H3, I mean, those might be infected or affected by mutations, but might not be, but those would be a little bit more, but we'd still be able to say that someone's infected with influenza A, which is helpful. And then we might say, oh, it's influenza A, but it's not showing up as a subtypable virus. Maybe this means it's something new.
Great. Next question from Cami Coons, and just a reminder, if you would tell us who you're with, that would be great.
Yeah, good morning. I'm Cami Coons. I'm a part of States Newsroom based here in Iowa. My question is, and I apologize if this is kind of a stupid question, but I think a lot of people's minds associate this because it has the avian flu with swine flu. And you've talked about comparing how this is similar to COVID or not similar to COVID outbreaks, but could you kind of compare it to the swine flu outbreaks that we saw several years ago?
Yeah, sure. So both avian flu and swine flu, if you're talking about the H1N1 2009, what was initially called swine flu, they're both influenza A. So they share that as a similarity, but they have different H and N genes, the hemagglutinin and neuraminidase. And so, of course, we're currently dealing with H5N1 versus H1N1. We know that the H1N1 swine flu, the 2009, became adapted for humans and then spread, and that's the virus that's still with us today that we get vaccinated against every year. The H1N1 is the version that came in 2009, whereas the H5N1 is still not yet adapted well to humans. So they're the same type, they're both influenza A, but different subtypes, and so far different ability to be transmissible to humans. Does that answer your question?
Yeah, I think so. I kind of just meant more so how it evolved to be transmissible into humans, but I guess you kind of talked about that with talking about how it mutates better in pigs to go into humans.
Yeah.
Any other comments from our panel on that question? No? Okay. Then Adriana has a follow-up, I believe. You're next, and then Apurva.
Thank you. So I wanted to get back to these tests and how they're able to detect the bird flu, because I know, unless they changed from influenza A to a different flu, we would still be able to detect it. But I wonder how sensitive are they? Because you mentioned that symptoms are the cases that we see are mild and that other cases may even be milder. And I wonder, the way that we've seen COVID tests not be able to detect virus because of the viral load. Maybe there's not enough viral load or whatnot. I guess I'm wondering how does that play, the sensitivity of these tests, and to be able to detect bird flu?
Dr. Bradley, that sounds like a great question for you.
Yeah, I think that's something that we're still trying to get data to understand better, are the viral loads in these patients. So a lot of that understanding also kind of depends on when these patients were sampled. And what we've seen in a few of these cases of conjunctivitis or respiratory symptoms is they have had fairly low viral loads by the time we've tested the patients. And that's made it more challenging to get the genotyping data from these samples. And that could be for a number of reasons. It could be exactly to your point that it's not replicating very well when it gets into humans. It could also just be, again, temporally kind of when this patient was sampled, that they're kind of on the tail end of their disease course. The virus is lower at that point. But I don't think we have very good data for that right now, especially the clinical presentations and characteristics. So I think that's something that really requires more investigation and is something that we do need to be alert and aware of when we're looking at the performance of these tests.
One thing I will add, I guess that's helpful to some extent, is having just come through the COVID pandemic, people are now much more aware of the different types of tests and PCR versus serology versus antigen. And so those general principles still apply. And Dr. Bradley alluded to that. If you come in early when you're really symptomatic and it's early on an infection where you have, really robust symptoms, you may be shedding more RNA and PCR might be more sensitive. Whereas if you wait a week and then test yourself, there's probably going to be fewer viral, fewer RNA for detection. So those general principles remain the same, and that's helpful to think about.
Okay. Our next question comes from Apurva Mandavili, I believe that is. I hope I got that right.
Yes,
And
it's
would
fine,
you
yeah.
tell us who you're with also?
I'm with the New York Times. So my question actually is a follow-up to the one I asked before about the tests. if you do have a test that, is positive for influenza A, so it's one of those, but you don't actually get a positive for H5 and you get negatives for H1 and H3, is that then a presumptive H5 or are there other possibilities? And then if you have to rely on sequencing, it seems like a couple of the cases that have been recent, they didn't actually have enough material to sequence, or at least only got sort of segments here and there. So what would you do in that case where you didn't actually have any kind of confirmation on what it is?
Yeah. So to answer the second question first, I guess they did have confirmation that it was H5 because for that H5 initial confirmation, it's just a PCR test targeted to the H5 gene. So they were able to confirm that H5 gene was
Sorry, I'm
H5,
asking a
but
hypothetical, not what happened already. I'm saying they didn't have enough material before, so it's possible that will happen again. So if you get a negative for H5 and there isn't material enough to sequence, and you have no information now on what this is, do you consider it a presumptive H5? That's what I'm asking.
okay. So I think that would require some epidemiological investigation as well. But from a clinical laboratory standpoint, we do have some assays that do provide subtyping as well. So it will say influenza A detected, and then it will tell you a H1 or an H3. And you're correct that there are situations where a lab could see a flu A detected, non-H1, non-H3. And that is absolutely a signal that this could be a H5 case that would require further testing, either sending out to the CDC or testing in the lab. Not that we have any other emerging pandemics right now of influenza virus, but if we did have some new novel version of H1 or H3 potentially where those historical H1 and H3 primers and probes have mutated, we could also get a result in that situation as well of non-H1, non-H3. Over the summer, the CDC did kind of issue a request to laboratories to increase surveillance for these types of flu A's that are non-H1, non-H3, and to have them sent for further sequencing and genotyping. So that has been something that's been increased, certainly in our lab, and I'm sure in Dr. Pritt's lab as well. Whenever we get any of these non-H1, non-H3s, which have happened in the past too, we will send those to our public health labs for further sequencing. Often there's just a mutation that causes the H1 or the H3 to fail, but it is an H1 or an H3. But we have been trying to maintain surveillance for these kind of novel influenza viruses with those patterns.
Okay, sorry, just a quick follow-up. Oh, God.
Oh, well, I was just going to say, to follow up on your question, there probably will be times where you get a non-subtypable virus, and then you try to send it for sequencing, and then there's not enough genetic material to give a confirmed answer. And then you're right, you would be saying, unsubtypable. But then that's where, as Dr. Bradley said, trying to get some epidemiologic information. Is the patient a dairy cattle, a farmer? Did he have exposure to dead poultry or livestock? So that might be helpful, but there will be cases where we just won't be able to give an answer.
Thank you. I just have a very quick follow-up to that. Part of the reason I was asking is because I know we don't have a problem with H7 and H9 right this minute, but if it's not a dairy worker and it's interaction with a wild bird, which we have seen now, for example, would you be able to tell if it was not H7 or H9 and that it was H5? Is there anything else you can do, basically?
If it goes to the CDC, they do have specific assays for H7, H9. They could try running those. Sequencing is also very helpful as well. But again, you're going to have those situations where there just won't be enough genetic material and you may just end up with a non-subtypable kind of result. So there may be some unknowns there, but I think the public health lab has a number of tools in their toolbox that can help try to solve the problems. And then they have their epidemiologic intelligence officers, EID, where they go and they actually do investigations to see what exposures the patient may have had.
Okay, thank you.
Thank you. Carter has a follow-up.
Yes. So this is one thing that I definitely did not know until the pandemic. I don't know anything about epidemiology. I didn't know anything really about this field at all until the pandemic and covering it at a wide scale. But one of the things was that flu shots were A and B based on trends that were happening. What were this year's shots, and do they offer any type of protection in a case like this, given that it is a type of A?
Yes, I have that information. So the 2024 and 2025 flu season, of course, we now have egg-based vaccines and then these cell or recombinant-based vaccines. And so they do use different strains, but they, like you said, they're based on the circulating viruses and the specific strains of those viruses. So we have an H1N1 and an H3N2 in both of them. And then there's also a B, an influenza B component, which the long name is it's a Victoria lineage like, and it's Austria strain, and it has a lot of numbers associated with it. But essentially, when we got vaccinated for influenza this year, you got vaccinated against the two circulating subtypes of influenza A, and then the one circulating subtype of influenza B. Does that answer your question?
Yeah, and then like, is this just something that's so completely different that it offers no type of protection in that?
You know, there's a hypothesis that the N1 component may offer some protection because we have a human circulating H1N1. And so if you have an H5N1, the N1 component of the vaccine may possibly provide some protection, but that is completely a hypothesis at this point. It's experimental, and I don't think we have enough information to say if our existing vaccines would offer any protection. I don't know, Dr. Bradley, do you have anything to add to that?
Yeah, I think most of that data at this point, Carter, is very in vitro and experimental. And I honestly don't feel totally prepared to speak on the literature at this point. But what I can tell you is that just because you get infected with one influenza virus doesn't mean you can't get infected with another. And particularly when we think about the folks who had really bad outcomes from COVID, these were folks who kind of had at baseline kind of decreased respiratory capacity, some chronic diseases. And so, I would say even if there were minimal protection to H5 provided by the H1, it's still an incredibly good idea for everyone to get vaccinated against H1 so that if H5 were to begin to spread, you wouldn't have folks who are potentially co-infected with H1 and H5 and having more severe outcomes from that situation, and just to sort of keep as much respiratory health as they can if they were to be infected by this novel H5N1 virus.
Thank you, Dr. Bradley, Dr. Pritt. We have a question from Lisa Krieger, I believe it is. Lisa?
Yes, thank you. I'm with the San Jose Mercury News out in California. We're dealing with a lot of this. Forgive me if you reference this. Is there genomic surveillance of the wastewater samples that all the public health departments now are collecting? It seems like, that could help with early detection of some of the emerging worrisome lineages.
So I am probably not very well versed to speak on this since I'm not in public health, but I know there have been some efforts underway. I'm not sure if it's specifically in California or not to do this kind of surveillance testing. But certainly we've seen the value of surveillance testing, not just for influenza, but for outbreaks of poliovirus, for monitoring for SARS-CoV-2 outbreaks. So, there is a role for doing this. And I think expanding that in these situations to find communities where this could be emerging that we haven't been monitoring in. There's always that risk, right, of if you're not looking for something, you're not going to find it. And wastewater testing gives us a really great way to look in lots of other places to see if we need to be more focused in doing surveillance for H5 in those areas.
Yeah, I'll add, Lisa, that's a great question. And it shows to my previous point that everyone is just a little bit more fluent in all these terms now. I don't think any of us would have been fielding questions on wastewater testing 5-10 years ago, but with the pandemic, I think there's more knowledge about some of these terms. I would say check with the California State Health Department. They probably have more information about specifically the testing they're doing. Like Dr. Bradley, I'm aware that some are doing it, but I can't say for sure if it's widespread throughout the different states.
Thank you.
So we have about 10 minutes left. No other hands are raised, and there aren't any new comments in the chat. So I would just open it up. Does anyone want to raise their hand for a final question of the panel? No? Okay, don't see that. Dr. Karcher, then would you like to make a closing comment here before we give some information about how to reach out if there are follow-ups?
Yeah, thank you very much, Robert. Once again, I want to thank all of you for joining today's briefing on the bird flu outbreak, and also for your great questions. Wow, you were really very much on point in terms of coming up with some great questions, and I hope we were able to answer them satisfactorily. Also want to thank my two colleagues, Drs. Pritt and Bradley, for providing some expert information about the virus and about laboratory testing. If our guests have any additional questions or would like to arrange a follow-up interview, please reach out to the CAP Media Relations staff. I believe we're going to post contact information for Angela Panettiere and Abby Watson in the chat, and right on cue, there it is. So please contact our media relations team if you would like some additional information or an interview. And thanks very much. Have a great rest of your day.
Thank you, everyone. Okay. Well,